Welcome to today's lecture on single-cell data analysis. In this lecture, you will learn about what single-cell RNA sequencing is and how it's different from the bulk RNA sequencing, and some basic principles of the data analysis, such as quality control, data normalization, and clustering of the cells based on the gene expression profiles. In the bulk RNA sequencing, the resolution that we are looking at it is at the entire tissues. So you take a tissue, extract the RNA out of all the cells within this tissue, and then sequence all of them together. When you do such sequencing, what you see in the end is an average expression of the genes of all the tissues, all the cells within this tissue. For the differential expression analysis, you take another tissue and then do a similar sample preparation, and then use some, after sequencing, use some normalization methods such as DE-seq2, and then look for the differentially expressed genes that show on average different expression levels between these two tissues. Coming to the single cell RNA sequencing, the resolution that we are looking at is actually at individual cells within these tissues. So if you sequence one tissue, even this tissue X here, you see that there are multiple many different cells in this tissue, because this tissue is made up of many cells, and then we see a different expression profile per each individual cell. For example, here in the cell 1, 2, and 3, there is an overexpression of gene A, and cell 4 and 5, there is an overexpression of gene B, and these last 3 cells, you see an overexpression of gene C. So there are always a set of cells that share a common or similar expression profiles, and some cells that don't share, that have a distinct profiles. And when you compare this to the bulk RNA sequencing, it's completely different. So here, what you see here is an average expression of gene A for all the cells within this tissue. But here you see an expression profile per cell. And similarly for the tissue Y, you see per cell a different expression profile. It may contain different number of cells here, but sometimes you see there are some cells that show similar expression patterns in tissue Y compared to tissue X. For example, this overexpression of gene A can be seen in tissue Y here, and also tissue X here in the cells 1 to 3. And there's also overexpression of gene C in these 3 cells here, which can also be seen here. And there's also co-expression of A and B, which can only be seen in tissue Y, but not in tissue X. And there's also some unique pattern in these 2 cells here, where there is an overexpression of gene B, which is not present in tissue Y. So there are always some cells that share, even among different tissues, some cells that share similar expression profiles, but not all. So the whole point of the differential expression analysis and the single cell RNA sequencing is to find out the difference between one set of cells against the other. So we are looking for the genes that are differentially expressed even within one single tissue. Differentially expressed genes between a set of cells, maybe there are these cells here on the top have a different expression profiles compared to these set of cells here. So this is what the whole point of this differential expression in the single cell. It's not just comparing between the tissues, it can be also within one single tissue. You are looking at how the expression profiles of individual cells are. The transition from the bulk RNA sequencing to single cell RNA sequencing is not so trivial. So there had to be a lot of new methods for library preparation had to be invented, and there are new methods for the sequencing, new methods for the quality control, even for normalization, clustering, and so on. So there are all new set of new methods, even bioinformatics methods, to detect these sensitive changes in the cells that we did not need for the bulk RNA sequencing. Let's look at how these single cell and bulk RNA sequencing are differ when you look at the sample preparation. In bulk RNA sequencing, a thin slice of the tissue is taken, and then an enzyme is added to break down the cell walls, and then all the unwanted DNA or RNA is rinsed out. So this can be, for example, ribosomal RNA that we don't want in the RNA sequencing because it's most abundant, and then you just sequence the ribosomal RNA. So usually people just deplete the ribosomal RNA, and then after that, the sequencing is performed on the whole leftover RNA in the sample. Coming to the single cell RNA sequencing, also start with the thin slice of the tissue, but this time before adding the enzyme to break down the cell walls, we separate the cells. So each cell is isolated to a different environment, and then after cell isolation, an enzyme is added to break down cell walls, and then a so-called barcoding is performed. So barcoding is kind of marking cells or transcripts within each cell with a different sequencing, so that in the end we will know which cell each transcript is coming from. So we will learn about barcoding in later steps in this lecture. And then after barcoding, we pull all the samples, all the RNA from the cells together, and then sequence them together in a common pool. One thing that needs to be considered, which is different in bulk RNA sequencing compared to the single cell RNA sequencing, is the biological replicates. In the bulk RNA sequencing, each tissue is a sample. So each tissue, that means all the cells within this tissue are sequenced together. That makes one sample. You take another slice of this same tissue, you may prepare the sample in the same manner as you did before, and then you make a biological replicate of it. So you can repeat it multiple times, and then you generate biological replicates. In the single cell RNA sequencing, it's quite different because if you take a different slice of a tissue, maybe it has cells which have completely different expression profiles. So you cannot just take another slice of the tissue and generate a biological replicate. This may happen that where the tissue is taken, the cell profile, the expression profiles of individual cells may differ. So that's why the concept of biological replicates is not similar to bulk RNA sequencing. So because each cell has its own unique expression profile. Let's look at this some important steps in this sample preparation, namely the cell isolation here and the barcoding, which are different from the bulk RNA sequencing. So let's look at for the cell isolation step. So in the past, the cells were isolated by humans by doing a manual pipetting. So a human takes a thin glass tube to suck up cells by maintaining a constant pressure in the tube, and then transport them into a new environment where they can investigate each cell separately. But this can be really time consuming because to isolate 1,000 cells, you need to repeat this process, manual process 1,000 times, which is time taking and also error prone. Sometimes you may get a cell, sometimes you may not get a cell, maybe multiple cells depending on how you maintain this pressure. So this is quite error prone. Nowadays we have luckily these automatic machines which do all this cell isolation, and one of them is flow cytometry. Here is how the flow cytometry works. So all the cells with this breakdown, which are breakdown from a tissue, are put into a saline liquid, which is here, and then this liquid passes through a really narrow tube, which permits only one cell at a time, but it's also really fluid enough to allow a high throughput. And the cells are quickly passing through this narrow tube, and they are collected in the end and then isolated in different environment in a plate. and then they are prepared for sequencing. But when they are passing through the cell, flow cytometry uses some lasers to investigate or probe some properties of the cells, so such as cell size and cell type. So, and they can also we can also use some fluorescent labeling, like these fluorescent markers that are attached to the surface of the cells, which illuminate at different frequencies of light. And depending on which cell types you are taking, so you can pre-sort your cells and then add a different labeling for each cell type, and then you can clearly see, okay, which cell types you're actually sequencing. And then the way that we measure this cell type and cell size is by these lasers here that are shot on each cell, and these detectors, so-called this forward scattering detector and this side scattering detector. And we will see how these forward and side scattering help us to detect the cell properties. First, look at the forward scattering. So the forward scattering is measured along the path of this laser. So the forward scattering is here. So it's exactly in front of the laser. The cells goes through it, and it just has a detector here. The laser shoots at the one cell at a time. And then when there is a diffraction of light, you see this on the detector, and if the diffraction is really high, then you see, okay, the cell might be of larger size. And the diffraction is low, then cell is probably of smaller size. And this kind of information is gathered here on the detector, the forward scattering detector, sorry. And the intensity of the forward scattering is actually proportional to the diameter of the cell. So here, what you see, a plot of here on the x-axis of forward scattering intensity and the y-axis side scattering. So let's first only look at the forward scattering. And the forward scattering is generally good for distinguishing between immune cells. For example, here what you see are lymphocytes and monocytes. So monocytes are usually much larger than the lymphocytes, and that's why you see the forward scattering intensity is much higher, and you see clear distinct two clusters of cells which are coming from lymphocytes and monocytes. And side scattering measures perpendicular 90 degrees to the laser. So the side scattering is somewhere here. So this is the laser path. This is where the cells are going through, and then the side scattering measures from here. As it's measuring from here, the intensity that you're looking at is much lower than the forward scattering. And this measures refraction or reflection of the light. So if you see, if the cell passing through the laser in front of the laser contains a lot of material in it, if it is really dense cell, then there is a lot of reflection because the light cannot pass through it. You see a reflection there. And if the cell is actually empty, if it is really less dense, then you see a refraction. The light can pass through it, and then it refracts. So based on these properties of reflection and refraction, you can measure the granularity of the cells. For example, here you see two different clusters of the monocytes and granulocytes based on the side scattering here on the y-axis. So the monocytes are usually much less granule, much less denser than the granulocytes. And this is what you see here. So the granulocytes reflect more light than the monocytes because they have a lot of fluid inside. That's how the cells are sorted, and you can also measure some properties. As I said, we can also use these fluorescent labeling for the cells. These are some kind of cell surface markers, which are put on each cell surface before you put the cells into the flow cytometry. And the naming convention here is a fluorescent labeling. So you can actually measure the fluorescent labeling as either positive or negative. If a fluorescent label is activated for this particular intensity of the light, then it's called positive. or if it is not, then it's negative. For example, here you see a plot where plotted against a CD4 marker and CD8 fluorescent marker. The CD4 is usually attached to the cells, CD4 marker attached to the cells of T helper cells, and CD8 is for cytotoxic T cells. And CD4 and CD8, all of them are usually CD3 plus, that means T lymphocytes. So all these two cells are subclasses of lymphocytes. And how to interpret this image here? So here on the x-axis you have CD4 scattering measurement, and here CD8 measurement. And when you see here CD4 plus, that means the fluorescent label was active, and you see a light for CD4 marker. If you see CD8 plus, that means the fluorescent label was active for CD8 marker. And that's how you measure. So here in this figure, you see three distinct clusters, where this cluster on the top bottom right is CD4 plus and CD8 minus. That means these are actually represent T helper cells. And here on the top, CD8 plus but CD4 minus. That means they are cytotoxic T cells, because these are the CD8 active on the CD8 marker. And there are some cells which are not at all active for CD4 or CD8. So these are cells which are a subclass of CD3, that means T lymphocytes, but not T helper cells or cytotoxic T cells. That's all about the cell capturing or cell sorting. And now let's look at the barcoding, the next step that differentiate single cell RNA sequencing from the bulk RNA sequencing. So the barcoding, so what usually is done is extract the RNA is extracted from all the cells, and then all of them are put together and then sequenced. That's what we usually do in the bulk RNA sequencing, right? In this case, we cannot distinguish which cells are which transcripts are coming from which cells. Let's just take an example here. We have exactly the same red transcript present in all of these cells here. When we sequence all of them together, there's no way that we can distinguish, because all of them produce exactly the same sequence, same read in the end. And then, yeah, there is no way to distinguish which read is coming from which cell. For this reason, what is done in the single cell RNA sequencing is adding a barcode. A barcode is a sequence that is attached in front of each transcript within a cell. So, for example, for these transcripts within this cell, we add a barcode GGG. But for the transcripts, for all the transcripts within, not just red, but all the transcript within this cell, we add a sequence in front, the 5' end as CCA. And then we can, when we sequence them in the end, we can easily distinguish that, okay, if a sequence is starting with GGG, it is, even if it's the same sequence, even if it's a red transcript, this is from a different cell compared to this one, which is starting with CCA. So what is done here is we have a plate with cells, with wells indexed by the barcode. So you have in each well one bead that contains hundreds of thousands of barcodes. And then you have each cell is thrown into this well. So when they're isolated, and then, yeah, once the cells are broke down, the barcode is attached to all the transcript within the cells. And then we can easily distinguish after sequencing. So the sequencing can be done in two ways. One is single sequencing, the other one is pooled sequencing. When we do single sequencing, each cell is separated, isolated from the remaining cells, put into a different environment, and then sample is prepared and sequenced as you do for the normal RNA sequencing. So each cell separately. This usually yields low throughput because you have only one cell. The material is all the reads that are coming from only one cell. Yeah. So, but this is how it is done. The problem with this is you usually have different capture rates for the cells, but there's also one more important problem is that is amplification rate, because we isolate cell into different environments. Each cell is amplified independent of other cells, and then you have different amplification rates in these different environments. Yeah, in this case, it's we need really clever tools for comparing samples across different cells, because yeah, each cell is a different sample and each cell is amplified at a different rate. The other way, more sophisticated way of doing single cell sequencing is, yeah, as I said, we can barcode the cells and then just pool them all together and then and amplify them together, because we know which we added the barcode, so we can just put them all together and then amplify together and then sequence them. So in this rate, the capture rate might be differ, but the amplification rates are all same. Then we don't have this problem anymore. But we need an additional step of adding barcodes. So let's look at briefly how the amplification methods that you already seen in the bulk RNA sequencing, and then we see how what we need for a special why we need a special amplification method for single RNA sequencing here. Usually the most common amplification methods that are used in RNA sequencing is polymerase chain reaction or strand displacement amplification. So these two here. Let's look at the PCR quickly. The PCR take a DNA, can be the cDNA generated from RNA, and then a primer is attached to the one end. So here it's the 3' end, and then after the primer, after this attaching primer, it is extended by incorporating one base by one base, and then extended until the end of the fragment is reached. Once it is reached, the one strand is synthesized, and then now you have two strands. Then we start with these two strands. We repeat the process in the next cycle. We get 4 strands. Repeat the process from 4 strands. We get 8 strands, and so on. So this is how the PCR is done. This is really exponential process. There's also another method which is called strand displacement amplification. So in this method, we also anneal the primers, but this time we start with denaturing of DNA. Then we take one of the strands, anneal not just one primer, but multiple primers. And then this time, the primers are extended until they reach the next primer, the start of the next primer. Once it reaches the next primer, it just displaces the primer, it continues, and the strand that we sequenced is just ripped off. And then on this ripped off strand, there are again new primers are attached, and then this continues, this process continues. This really yields a high throughput. The only difference between STA and amplification is two differences. One is this displacement here, and one more advantage of STA over the PCR is when we produce a complementary strand here, we need to denature this to separate them and then to do a second cycle. So we need to always heat it up and then denature and then amplify each strand separately. But in the STA, we don't need to denature the samples. You can just continue it. The strands are displaced by this extension. This is also really high throughput, and both of them are usually works well. So both of them can produce a really high library size, and but the But the problem here is both of them cannot produce really a complex library. So we'll see what the complex library complexity is. So here's an example here. So you have a cell, let's say, just assume that you have a cell with one transcript each. So the one transcript is really low because you don't have multiple copies of the transcript. Then the problem here arises when you have such really low number of transcripts. The PCA or SDA, PCR here, so there's a typo. PCR or SDA works well. No, sometimes works well because sometimes they pick up this fragment, one of these fragments. For example, in this case, a red transcript is chosen and then amplified it multiple times, but these methods may miss this blue transcript, and then they just do not sequence because these two transcripts are really low in number. So if the high number of transcripts in the cell, you can sequence this, but if there are not many blue transcripts there, then it's then sometimes the amplification may not pick up this transcript, and there's nothing amplified. In this case, then we get a wrong conclusion that there are many red transcripts in the cell, but not blue transcripts in the cell. But in reality, there are equal number of red and blue transcripts. This cannot be. Yes, this is one of the problems that this PCR and SDA cannot solve. And this is what library complexity is. So you need to produce a library, the amplified library, that is actually equally good as what you have in the cell. So in theory, you should at least see, if an amplification is good, you should at least see at least one copy of blue fragment here. Then you have really many different unique transcripts after amplification. So if you have many different unique transcripts, that means your library's complexity is high. And to solve this problem, especially in the single cell sequencing, new amplification method was invented. That's called MALBAC. MALBAC stands for multiple annealing and looping based amplification cycle. So it starts similar to what you've seen in the SDA, extend and displace. So you start with a DNA or cDNA, and then denature one of the strands, do as we did with the SDA, but this time we anneal the primers. We do one time extend and one time displacement, and then we produce these 3 fragments, and then we stop there. We don't go further. We don't add primers here and then extend these, and then we don't repeat this process. But once you generated with the initial primers these 3 complementary strands, these 3 complementary strands are again considered to do another round of amplification where you generate the opposite strand of these complementary strands, which is usually this strand. But this time we have primers that are complementary to the primers that we attached here. So in this case, we also run one round of amplification. Then we generate such fragments that have complementary primers at the ends. And then what happens here is these complementary strands, complementary primers are forms loops so that these cannot be amplified further. So you get these products here. These are the end products. And what the advantage of this is, so you can take all your transcript from the cell and try to do several steps of this extend and displace in the beginning, so that you try to collect as many unique transcripts from the cell as possible. And then once you have reached a a decent number of unique fragments or decent number of distinct transcripts, then you start with a PCR, and then you do a PCR on these fragments. So in the beginning, in the other methods, you probably just have one such transcript, which is amplified multiple times, but this time you try to get as many unique transcript as possible, and then you give it to PCR, and then we amplify, and then in this way there's more chances of getting unique expressed transcripts from the cell after amplification. So there are some issues with amplification, because amplification is good really for the sensitive detection of the transcript in the cell, but it can also cause some problems. These problems are the following. So one problem, most importantly, let's assume a cell with 2 red transcripts and 2 blue transcripts, and if you have 2 red and 2 blue, and if they are amplified, and we get 6 red and 3 blue. This is just because of, as I said, how the fragments are chosen. You may choose the amplification process may choose a red multiple times, then we have multiple red fragments here, but only 3 blue fragments, less number of blue fragments. In this case, we cannot really infer how many how many number of red transcripts were there in the cell, and also how many blue transcripts were there in the cell. If we just deduplicate all of them, we just think either we think there are 6 transcripts, red transcript, 3 blue transcripts, or we just say, okay, all of them are exactly the same sequence. Maybe there are only 1 red transcript and only 1 blue transcript. And both these assumptions are wrong, because we have 2 red and 2 blue transcripts. So to overcome this such amplification problem, this overcounting or undercounting this actual number of the real number of transcripts in the cell, what we employ here is so-called unique molecular identifiers. The unique molecular identifiers are, again, they are similar to the barcodes that you see in the cell barcodes, but this time they're also really tiny sequences, maybe 5 or 6 nucleotides of sequences, which are attached to the fragments. So the fragments are simply these transcripts contain already a cell barcode, and then these UMIs are attached in front of the cell barcodes here. And these UMIs can be used to identify your transcripts uniquely after amplification. So here, what you can see, after amplification, we see that there are 2 red transcripts with a UMI that is attached in front, which is pink here. There are 4. The remaining 4 red transcripts contain a blue UMI. Same here, there's a pink UMI attached to blue, which is amplified once, and then a green UMI attached to blue transcript that amplified twice. And now you can, when you count them, you still get the number of reads. What you get is 6 and 3. But when you group the reads by gene, the transcript and UMI, then you get actually the actual number of transcripts that are present in the cell. So the pink ones, for example, take the UMIs here. There are 2 red reads with pink. 4 red reads with cyan, and so on. And then when we deduplicate them based on the UMIs, you just see for red read there are 2 UMIs. For blue read there are again 2 UMIs. This indicates that we have exactly 2 unique reads for red and blue. That means we have originally in the cell 2 reads for red and 2 unique reads per glue transcript. Yeah, so that's how these UMIs can be used to deduplicate the reads. So you can so the UMIs are actually can be used to deduplicate at the transcript level. For example, here there may be sometimes UMIs that are the same UMI can be attached to different transcripts. For example, the pink one attached to red and blue, but we don't have any problem with this. We are safe because the sequence are different. The sequences of red and blue, red and green are different here. The red and blue are different here. So even though the UMI is same, we can distinguish by the sequence. If the sequences are same, we can distinguish this by the UMIs. So UMIs, with the UMIs, you can distinguish this, transcripts at the transcript level. Now the question is, can we overcome this problem that we don't need to, we don't want to have this same UMI attached to different transcripts? Yeah, to find out this, we have to know in advance how many mRNA transcripts are there in the cell. So this is anywhere between 100,000 to 1,000,000 transcript in a mammalian cell. So this means we need at least a UMI of length n, where the 4 power n is equals to number of mRNA transcripts that we expect in the cell. So that means because 4 power n, because we have 4 different nucleotides to choose from, A, C, G, and T. And then when we solve for this, you get somewhere at I think when I solved it somewhere, it's 8.3 or something. And then if this is not a whole number, then when you round it, you get 9. So that means the n is 9. That means you need a UMIs of length 9 to generate unique number of UMIs, unique number of 100,000 UMIs. Yeah, if you are going for 1,000,000 unique UMIs, you need somewhere, I think 10 or 10 power 6. So these 9 and 10, the length, the UMIs of length 9 and 10 is unrealistic in the real world setting. So these are really expensive to produce such longer UMIs. Hence, yeah, this is not usually done. Yeah, people go for usually 5 or 6 in general, mostly 8, let's say. So let's look at how these UMIs are generated and why it is important to generate them in this manner. So to generate the UMIs of length n with an added distance of b, so n is equal to 5, for example, here, and added distance of b, 1. So that is the added distance says how many nucleotides we need to change to generate a different UMI. So the difference between the first one and second one is 1. For example, the A is replaced here by C, and G is replaced by C. So each one is unique, but there is only one edit distance among all these UMIs. In such a way, you generate 4 power 5. So that is about 1,024 UMIs. And another example where N is 5, B is 2. At this time, 2 nucleotides are replaced or changed each time. So AA is changed to CC here, which is changed to GG here, changed to TT here, and so on. In this case, we generate 4 to the power of 5 minus 1. That is 512 UMIs. That's half of this. So with increasing the edit distance, you decrease the number of UMIs, but the edit distance is actually good because these actually can edit distance can guard the UMIs against the sequencing errors. For example, if we have an edit distance of 1 here, let's just consider these two first two UMIs. And if there's a sequencing error in the second one, and if it, instead of sequencing it as a C, let's say it has a here, then you have both this UMIs exactly the same. Then when these are attached to the transcripts, there's no way that we can distinguish them. And if you have a larger edit distance, let's say 2 or 3, then we can at least assume that okay, there is if there is a C this instead of A, then maybe then you can say okay, it might be, there's a instead of C, then you can say okay, it can be an A which is sequenced, when it can be a C which is sequenced as A, or it can be a G which is sequenced of A. So you can at least have guess what it can be, because the bioinformatics tools also take care of such sequencing errors even in the UMIs. And usually the B that is used in the scRNA, the single cell protocols, is about 2. The UMIs that are selected must be unique to the genome that you're mapping to, or the sequence that you're sequencing from. So if the UMIs can randomly match to some place on the GM and the genome along with your transcript, so we attach UMI in front of the transcript, and this if the UMI and transcript together can match to the genome uniquely, then it is a problem. Then you actually mapping your read to a different position that than what you actually wanted to. And that's why these UMIs should be really unique, and they should not be accidentally mapped to the genome that you're sequencing from. And that's why these UMIs are designed to be genome specific. So you see a different set of UMIs for human than mouse, for example. And this the edit distance also must not be too small. So 1 is never a case, because then you don't guard for the sequencing errors. So you can imagine these UMIs as a random salt that is just thrown, as we also seen, just thrown to this plate of wells. And then they are unique enough to distinguish at the transcript level. So that's why we count the transcripts only. So we deduplicate the UMIs at the transcript level, and then we can, in this case, then we can get a good estimation of transcript abundance after quantification. There are still some issues with the UMIs, which one of them can be solved, one of them not. So let's look at these two issues. This hypothetical one, where the pink UMI here captures 2 reads. So the pink UMI is attached to read red, transcript red and transcript blue. This is not a problem, as we've seen before, and when I introduce the UMIs, that's not a problem. We can still do duplicates at the transcript level, because these 2 transcripts are different. Even though the UMI is the same, the transcripts are different. It's not a problem. This can be solved. But the hypothetical example 2 here is a problem. So where the pink UMI is attached to the red 2 different unique transcripts. In this case, it's hard to distinguish after sequencing which read is from which red transcript. So in that case, it's we call this as UMI collision, and deduplicating red will cause problems. It's hard to deduplicate. There's no way to deduplicate them. And that's some issues, and that's about UMIs. And now just recap again the barcodes, cell barcodes, and UMIs together. So what's done at the beginning? First, put all the cells in the wells where there is, in each well there is a bead with unique cell barcode. For example, there's a well with only GG. So all these three Gs, and then that cell is put into this well, and then after breaking down all these GGs are attached to all the transcripts within this cell. So all the transcripts contain GGG after you sequence. And then after that comes the UMIs. No, sorry, this is another cell here which contain which is attached with a different barcode, TCT, and all this transcript in the cell, in this green cell, has TCT attached to it. Then comes the UMIs, and UMIs are added to the cell barcoded cell, cell barcoded cells. Yeah, the barcoded cells, let's say. So there may be same set of UMIs for these two cells, because we can already distinguish cells by the cell barcode. Now we just throw all this same set of UMIs, and then these UMIs are attached to, hopefully uniquely attached to this to different transcripts. In the end, now we can distinguish cells by this barcode, cell barcode. We can distinguish transcript by the UMIs, and that's how we can actually achieve sequencing at single cell level compared to what we've done. The bulk RNA sequence, we just put all of them together, sequence them, and then we get just an average expression of the tissues. But here we can simply distinguish each cell with the barcodes and UMIs, and then we get expression levels of transcripts at cell level. And now after sequencing, what you get is so-called count matrix. So we sequence all the cells together, deduplicate them, you just extract the barcodes and then just distribute to the reads where they are coming from, and then we build a so-called matrix, the cell matrix, where we have count matrix, where we have all the cells on the x-axis, sorry, all the genes on the x-axis, all the cells on the y-axis here. So the columns represent cells, the rows represent genes. Yeah. So here we see that cell B has a gene 1 that has 4 transcripts. The gene 2 in cell B also expresses 4 transcripts. Gene 3 in cell C expresses 4 transcripts. That's how you read it. And then you don't directly consider this matrix for the further analysis. So before using this count matrix, first what is done is to so-called quality control, which is filtering this count matrix, because these count matrices have a lot of background noise, which sometimes you have really cells that are not well captured. Sometimes there are genes that are not detected because there's not enough transcripts in a cell of particular type. So in this case, we do filtering based on the column, that is based on the cells, or based on the row, which is genes. So what we do here is we take all the cells and then compute the library sizes. The library sizes is simply summing up all the counts, the read counts. So here 1 + 1, 2, 4 + 4, 8, 2 + 4, 6. Oh, okay, so this is not correct. It must be 6 here, not 8. And then 3, 3, 1, 7, and then 8. So that's how it is done. You compute the library size per cell, because each cell is a library, different sample. Yeah. And then now we detect, then we see at each threshold how many genes we detect per cell, right? At greater than 0, at least 1 read per library. In this case, we see for gene for cell 1, cell A, we detect these 2, but not these 3 because it's is equal to 0. For this, for B, there are 2. For C, 2. For D, there are 3 cells detected. For E, there are 3 cells detected. And we do also at different thresholds at 1, 2, 3, and so on. And then, yeah, we have such information here. And we do similarly for the rows. That's along the genes. So we have genes here, G1, G2, G3. Now we count a number of transcripts per gene among all the cells. So for G1, it is 10 here. The sum of these reads, and here it's 12, and here 4 plus 3 plus 2, it's 9. And that's what the number of transcript per gene among all the cells. And now we see how many cells we can detect at different levels of the gene expression. So we see here when you say greater than 0, cells detected greater than 0. So we see here there are gene 1. This is not detected. So we get 1, 2, 3, 4, and it's here. For gene 2, all of them are greater than 0. We can detect all of them, which is 5 cells. For gene 3, we can detect these 3, which is here. And then we do this for different thresholds. And then we sensitively choose, depending on how many cells that you expect. If they are really, we just plot it, and then we see whether they are extreme outliers, and then see, okay, these are not fitting into the distribution of the majority of the distribution of the cells or genes, and then we just throw this out by some cutoff. So that's how the cells are filtered. That's a common quality control that is done usually before proceeding. And let's look at now the normalization, because after filtering, we normalize the data. So as we did for the bulk RNA sequencing. Just recap what we did for the RNA sequencing. So you have 2 different conditions. So this is healthy tissue. This is a diseased tissue. And then you have the same set of genes for these 2 tissues, and then now compare how this gene R is expressed between these 2 different tissues. So here it's overexpressed, and gene B is underexpressed compared to the diseased tissue. That's how we see, and that's why these are the factors that are important for us. What type of the tissue it is, or what type of the sample it is, or what the difference between the gene expression across the samples. Factors that we don't care about is how big are the samples, whether these are sequenced at different depths, and how much samples are amplified. We don't care about it because we cannot solve this amplification problem there. So we don't care about the amplification or the how big are the samples, because the samples are anyway in the end normalized by some normalization method, for example, with sec2 by the, where we take the size factor and then we normalize them all to the depth of the at which they are sequenced. The factors that we might care about when we do such differential expression analysis or normalization at least, is the type of sequencing we use. So this is one of them. So it may influence or may not influence. So everything that you think that influences your experiment is one of the side factor. And this can be, for example, if you sequence exactly the same sample, single end or paired end. This is kind of a technical factor. So you're using a same machine, but you're doing 2 different types of sequencing, a single or paired. This may influence how the samples are, how the distribution of the reads in the end. So this is something that we may need to incorporate when we normalize the samples. And the purpose of the normalization is just to keep the factors that we care about and then just ignore whatever we don't care. In the bulk RNA sequencing, you usually see high coverage. Why? Because we have cells, all the cells that are common to that are coming from a single tissue. So in this case, for example, gene A, we see 100 counts from tissue 1, and this tissue 1 contains, let's say, 1,000 cells that only some of the genes are expressed, some of the cells that have this gene expressed, and then on with some of them together, then probably 100 reads that are coming from gene A. Similarly, for gene B in the tissue 1, there are 45. So we usually see high coverage because these the counts are averaged over all the cells in these tissues. So that's why the median of gene expression is high. In the single cell RNA sequencing, we usually get a really low coverage because this T1 here, the tissue, is now not anymore one single column here, but it is split into 1,000 different columns. So each cell is its own column. But gene 1, so in order to get to T1 here, you probably need to sum all these cells, then probably you get 100, but per cell you have really low number of counts, so no low number of reads. And that's a problem because the median of the gene expression is sometimes 0. So you may see sometimes in some cells there's no genes are expressed. So when you take the median, this might be simply 0. Why that is important? Because if you recall the DSEC normalization, what we take first is this denominator, which is a geometric mean among all the counts here. So take geometric mean from this, and then divide each count by this geometric mean, and that's what we do. And then once after we're dividing, we take all these divided numbers and then go through these rows and take a median. That's what the size factor for each sample. That's what here it's done, the TSEC for the bulk RNA sequencing. But here, as we have zeros here, the geometric mean can be 0, and then also this median can also be 0. So this is a problem because, yeah, in math we cannot divide by 0. So we cannot do it in practice. So that's why what we have to invent a new normalization method. We'll just look at this problem again. What happens in the cell? So this is the case where we see only some or at all no reads from some cells is called some so-called gene dropouts. So just let's look at this example where there are 2 cells A and B of exactly the same type. So represented by this gray color here, that means they are from same cell type. In theory, they must have exactly the same, not exactly, at least reasonably same cell gene expression profiles. In this case here, we see exactly 2 number of red transcripts that are there in the cell and 1 blue transcript in both of them. Yeah. But when these are amplified, we see 6 red transcripts and 3 blue transcripts from cell A, but only 6 red transcript, but there's no blue transcript from cell B. So this is called gene dropout. This transcript is completely dropped out. This never amplified when we never see the gene that corresponds to this transcript in our analysis. And this is usually the problem. This occurs usually in the single cell RNA sequencing, either because of the poor mRNA capturing efficiency, or it might happen if there are low number of transcripts present in the cell. So let's say if there's only one red transcript or blue transcript present, there are many red transcripts. You may pick up one of the red transcripts and then amplify them. But yeah, as we discussed before, sometimes this may missed and then there's no blue transcript present. And this actually leads to our count tables, what we've seen here, with a lot of zeros. So we see this matrix of cells and genes. And this matrix usually, in reality, in the single cell RNA sequencing contains a lot of zeros. This is a problem. And this leads us to a model, a count model, which is called zero inflated model. So normally the gene expression ideally follows negative binomial distribution. And this is also the assumption that DSEC makes, that DSEC assumes that the counts follow the negative binomial distribution, and that's how it models the counts. But yeah, as we have a lot of zeros there, because of these gene dropouts, we get a mixture model of two peaks. One is centered at 0, the other one's elsewhere there is expression levels. So we get such two peaks where something like this. This peak is usually much higher, and this is somewhere 0, and this is somewhere, let's say, I don't know, let's say counts of 100 or something. Yeah, and this is usually broader because there are different types of counts. And this is not following usually this negative binomial distributions. And it's also, we cannot ignore the zeros. If, yeah, in this case, if the gene is not expressed in reality, it's still a differential expression. Yeah, that's like what people usually do. They in their tools they incorporate a pseudo low number, really like a constant, let's say 0.01 to all the counts, and then this can deal with this these zeros here. What we have, this problem, this divided by 0 problem, because here now we have everywhere 0.01, 0.01. Now we don't have any problem that's divided by 0. We can still deal with this, but the problem is it's not an efficient way of doing it, because you are actually you still over or under count these expression levels. The normalization is not exactly what you really want, because you have a lot of zeros in the count table. And that's why there are different methods that are developed for, especially for the single cell RNA sequencing. The scRNA is one of these methods that does a really fancy modeling of to normalize the RNA sequence, the single cell RNA sequencing data. Let's look at this method. So what it does is takes the strand first, take all the cells, and then computes for each cell library size. The library size is simply number of reads that you see divided by the whole number of the number of reads. So this may contain 10 reads. Just an example, let's say, okay, to be realistic, maybe 10 reads, and this may contain like 6 reads. This may contain only 1 read. So in reality, this is in factor of thousands, so 100,000, 600,000, let's say 10,000, something like this. So you compute the library sizes, and then after the library sizes are computed, we take an average, and that's so-called pseudo reference cell library size. Take average of all the library sizes within this all the cells within our single cell experiment, and this gives this pseudo reference size. With this, after computing these reference sizes, library size, the cells are ranked by by the library size. So this may get like, this may be rank 1, this is probably rank 2, because this has 10, this has 9, and this is rank 3, and this is let's say this is rank 4, and so on. Now all the even ranks goes, the odd ranks goes here, the red ones goes here, all the odds, this 3, 1 goes here, and this 2, 4 goes here. So all the evens goes here, all the odds goes here, and then they're all put into a ring, a so-called ring, and in a sorted order. So like this, in the descending order, and then this is simply a cycle, and they are just put on this ring. And now comes the normalization part. After they are placed on the ring, we choose a cell, K. The K defines how many cells that we want to choose at a time. Yeah, so this is called pool. So the K here is 3, for example. This green one is a K, so this is a pool of 3 cells. And there's another one here, a blue one, which is full of 3 cells again. So our K is 3. We choose a windowed approach, so we move our window 1 cell at a time. Our K moves, so our window moves from green, and then go to the next cell, blue, and go to the next cell, red, and so on. And while we move on this window, so for this each pool, sum all the library sizes for this, and then derive a size factor from the pseudo size factor that we calculated before. So for these 3 cells, so this is, let's say, theta 1, this is theta 1 here, this is theta 2, this is theta 3, and so on. Yeah, so for the second one here, for this ring, this green pool here, you have theta 2, theta 3, and theta 4. What we do here, we calculate size factor based on the library size of theta 2 plus theta 3 plus theta 4 divided by the theta ref that we calculated here, this one. Yeah, and we do this all these all the pools, and after calculating the size factor, and as they are overlapping, we see the same ring multiple times. For example, this one here, theta 4, 2, 3, 4, theta 4 can be seen in 3 rings, and theta 3 can be seen in blue and red ring, as blue and red pool. And we take an average, we build a linear model based on these size factors of these cells here. And then this gives a size factor for this particular cell. So linear model is built like alpha times the first size factor here for this pool, beta times the second size factor for this pool, and then gamma times size factor for this, because this cell presents in all these 3 pools. And then now we do, yeah, all the size factors are computed for each cell, and these are used for the normalization. Yeah, so this is how the strand normalization works. And now after normalization, what we have is, as I said, the count matrix that contain thousands of cells and tens of thousands of genes. And that's about a matrix of tens of thousands of dimension with thousands of observations. So the observations, cells are generally called observations in the single cell terminology. Our aim is to find groups of cells or which can be clustered together based on the expression levels of some of these, a subset of these genes. Yeah, And as we already seen in the beginning, the introduction slide for the single cell sequencing, some cells have similar expression patterns, but some actually have very distinct expression patterns within the cells. And we use such information from the expression, these genes that share different or distinct expression patterns, these highly variable genes, to cluster these cells or group these cells based on the genes. And yeah, once we group the cells, we want to visualize them, and and how this is how we represent it. So we take the count matrix. We can visualize this by simply taking these coordinates here, so the counts here. So what we see in this is 3 genes and 3 cells here. The RPV here is the cells, and here is the genes. Oh, that's bad writing. Okay, is cells and here's genes. And now we want to visualize the cells based on the expression of the genes, right? Now so our coordinates are, our axis are in this case genes. So we have 3 genes here. That means 3 axis. So it's 3-dimensional plot here. And now we are visualizing R here. R has on first dimension 1, second dimension 1. This is 1, 1, and that can be placed here. For P, 1, 3, 3, which is 1 here, 1 here, 3 from this dimension, 3 from G2, 3 from G3, and this is somewhere placed here. And same for G, V, cell V. And we can now see in 3D, okay, the cell R is somehow far away from the cell V, because you can clearly see this is closer in one of the dimensions than this one. This can be visualized, when we have such 3-dimensional, or in 2-dimensional, it's much simpler. But the problem here is we have in this example only 3 genes. But in the real world setting, it's not just 3, but it's actually 3 followed by 4 zeros. That's about 30,000 genes. And human mind cannot perceive dimensions more than 3, or at most 4. So we cannot see, or we cannot even draw such 30,000 dimensions. So we need to find a different way of doing it. So instead of plotting them directly, we do distance matrix based on the Euclidean distance among these cells from the gene expressions, and then we compute the Euclidean distance here. We create a distance matrix, and from the distance matrix, we can plot them. But as I said, it's possible to plot many dimensions. And then from this, what we take is what we do is a dimensional reduction. So we can use something like PCA to reduce from like tens of thousands of dimensions to 2-dimension, because 2-dimensional is easy to perceive. So the main constraint here is we have to preserve the same topology that you have in the higher dimensions, also in the 2 dimensions. For example, let's just take this example where you see that gene cell A is far away from cell D, which is pink here because there's no direct connection, but it's relatively closer to cell B, this blue one. There's a direct connection here, and this must be preserved when we reduce dimension from 3D to 2D. So, and that's how a good PCA should look like, a dimension reduction look like. So we have a direct link between red and blue here, with A and B, but not A and D, because there's no direct connection here. So the topology should be preserved in the 2D. So let's, before looking at how this dimension reduction is done, you will learn more about the dimension reduction in the next lecture, about the PCA and so on. So we are not going any further details. We are not going to details about this in this lecture. We will now learn more about the clustering, because we've seen in the beginning that even for one single tissue, what we are going to look at, all this differential expression, can be also within one tissue. We are looking at the cells that have different expression profiles based on these gene expressions. Yeah. So all these cells, for example, these purple cells have an expression profile where there is always gene A is overexpressed. These yellow cells have gene B and C and are normally expressed at moderate level, but gene A is underexpressed, so downregulated. And these green cells have gene B as overexpressed. So you can really see this when you look at individual cell profiles, but when we just take all the cells together, we see a different distinct expression profiles. For example, here, see an expression of gene A, either high expression or low expression. So you see two different peaks here. Same for the gene B, you have a moderate expression or a higher expression, and for gene C, the expression doesn't change across all the cells in this tissue, and you see a constantly same expression. And these expression profiles can be used to define what these cells are. So these cells can be seen as, okay, if there is a purple cell, this must have always a higher. If you see a higher expression of A and moderate expression of B and C, then this must be a purple cell. If a higher expression of B and low expression of A and a moderate expression of C, that must be green cells. And yeah, if you see different expression profiles, that must be yellow cells. So that's how we can do this. But you can also look quickly at a video that we provided along with this lecture, where we use select gene visualization, which is one of the popular visualization to visualize such clusters of such cells based on this gene expression. So you can just check out this video. You can also install select gene on your computer with this pip, and then you can launch it with the publicly available data sets, this PBMC3K, and then launch it locally, and then you can also do it yourself. Yeah, just check out this, and then you see such interpretation of the cell expression levels. Now coming to the, based on this such different expression profiles, we can cluster the cells. So we project them into 2D, and then we see such clusters. So each dot here represents a cell, and the clustering is done by one of the clustering algorithms. So we will see in a moment what briefly about the clustering algorithms that are commonly used. And these, based on the clustering, these dots are colored, and different colors represent different clusters, represents possibly different cell types. And these cell types can be discrete. For example, they represent different cell types, but to mark which cell types these are, what we need. So to annotate that, okay, this is neuronal stem cells, this is astrocytes, whatever, this is oligodendrocytes. We need to know first so-called marker genes. So we need to find out the genes that are highly variable in these clusters, and which usually are the highly expressed, and also which are really most important genes for such particular cell types. And we can find out such genes by looking at the expression levels, and then we can quickly say, okay, these, if we see a gene, let's say, I don't know, gene A, which is usually highly expressed in the astrocytes, so that must be in this cluster, must be astrocytes. And we see, okay, gene B, which is highly expressed in oligodendrocytes, okay, then this cluster must be oligodendrocytes. We find such marker genes, and then yeah, and define these cell types. And after defining cell types, what we can do also is infer the relationships. So that's lineage. The relationships is something like, okay, how these neuronal stem cells, starting from neuronal stem cells, we can go to neuronal progenitors, and then it can lead to mature neurons, or can start from stem cells, go to glia progenitors, and then oligodendrocyte progenitors, which ultimately leads to mature oligodendrocytes. Such lineages that we can also infer from this relationship, from this clustering. And this can be done by these lineage trees, which are considered by taking the entropy of the clusters and the proximity of the clusters. If the two clusters are closer, that must be like, okay, this cluster must be from here. If so, we can assume that common progenitors cannot be directly related to clear progenitors, because yeah, there are other clusters which are in between. So we take the proximity. We take also entropy. The idea, the motivation behind this is it is assumed that the clusters, that the cells of stem cells, the ones that actually differentiate to other cell types, have higher entropy, because they tend to give rise to or differentiate to different cell types. That's why you see that generally neuronal stem cells have a higher entropy compared to astrocytes or oligodendrocytes, because they are mature cell types, and the entropy must be longer. The entropy, by entropy, I mean here is the expression, the gene expression profile. So here you see a noisy expression profiles, but here in the mature cell types, you see clear distinction of the gene expressions between the dendrocytes and astrocytes. This information can be used to yeah, infer the relationship between these clusters and how these cells are differentiated. The clustering can be seen as hard clustering or soft clustering. So hard clustering is something what you see here on the left. You really see a big spaces between the clusters where the cell types are well defined, and this, yeah, you can clearly distinguish which clusters represent which cell types. There can also be soft clusters, so-called, something like this, where the cell types are just intermingle in one to another. And there's hard to say that these two are different cell types, because they are actually, yeah, really close to one another. But this type of clustering is sometimes actually very useful to solve this problem of lineage, inference lineage. Because in this such soft clustering, what you see here is how the cells are actually matured. So how the cells of one type of cells are differentiate to other type, and differentiate to other type, and so on. In the end, they form some mature cell types. So this type of clusters can be used. For example, for the red blood cells that are usually come from the reticulocytes. So these cells can be seen, but in this development process, we also have intermediate and rare cell types that we normally don't see unless we do a single cell data And then when we see such soft clustering, we really see that, okay, they are not discrete like here, what you see on the left, but they also can be also transition. So, and this changing over times from from stem cells to mature cell types can be seen in such a clustering only. So let's look at now how to perform the clustering. So there are different ways of performing clustering. One is k-means, the other one is k-medians, and they are kind of similar. And there are other types which are hierarchical clustering and community clustering. So the both of them are kind of hierarchical clustering algorithms. We will see each of them on each how they are performed. So first let's look at the k-means clustering. So we have all the points. So each point is a cell here, and then these cells are put on the this 2D plot, placed on this 2D plot, where we first randomly select k positions. So in k-means, we have to know, or we need to provide to the algorithm how many k's we expect. That means how many clusters we expect beforehand. So we should know this by our data, and then we say, okay, from this particular tissue, expect there must be at least 7 clusters that I expect, and then the k is 7. So in this case, our k is 3 in this example, and we place 3 k's at some random position. So you choose one of these cells as your k. And then after choosing a cell as a k, and then compute the distances from all the cells to k, and then assign each cell to this nearest k. So that means now we actually have a coloring of each cell to its nearest neighbor, nearest k. And then once we put all these together, like this colored here, group them together or assign to the nearest k, we move our k to the mean position, because instead of having now we have all these reds here, which are the nearest ones to this particular k, and then we take all the mean positions and then move our k to wherever it fits. Same done for the yellows here, same done for the blues here, and then we move our positions of the case, and then we iterate this process again and again until the k doesn't move, the positions of the means doesn't move at all, and that's where we converge. And this is how the k-means clustering is done. Then we have desired number of clusters, k number of clusters here. An alternative method is k-medians clustering, where instead of using mean position here of the cells at each iteration, we use median position. So the median position can actually take care about the is less influenced by the outliers, because you are looking at where your data is mostly concentrated, where your cells are mostly positions. So if there are a few outliers that are shifting, that are really far away from the remaining group, if you take the mean position, this may shift it, but actually the majority of your data points are somewhere here. That's why, yeah, in this case, the k-medians works well. And then there's hierarchical clustering, which is simply you need a distance metric for this. So take the distance matrix, and then from this distance metric, you take 2 nearest ones, merge them together, and then now again take the next nearest ones in this after merging, and then merge them together, and then take again these 2 ones which are near, and then merge them, and so on, and until you end up with only one single cluster. So in the hierarchical clustering, you really don't yield the clusters that we see in here, as in the K means that defined number of clusters. The clusters here are based on your tree. So how you actually define cut your tree. So because these are hierarchical, you see, okay, A and B are together. Maybe if you cut your tree here, A and B are together. But if you cut your example tree here, it's C, D, E, F are together, and then A, B, C are together, and so on. So this is simply what you see here is a hierarchy of clusters. And another clustering algorithm is a Levine clustering algorithm, which implements the community clustering. So this is based. This is based. You start with one of the connected graphs. So maybe this is generated from distance matrix. And this in this clustering algorithm, we first start with a connected graph, and then each node gets its own cluster in the beginning. So each of these colored nodes gets its own clustering. So this round circle defines the cluster. So in each cluster, you have at the beginning, in the first iteration, only one node. And now what we do here is, for each node, we compute so-called internal and external links. So the aim is to maximize the internal links and minimize external links. Yeah. So for example, for the pink here, we have one external link, two external links, three external links, four external links, and there are no internal links. So there's no other node in this cluster. That means there are zero external internal links. Now we try to maximize the internal, this one, and minimize this one. So we do this by probing, okay, what happens if pink absorbs purple? So if you just put these two together, right? Then we put them together, and now we count again. There are one external link, two, three, four here, and these two together are actually going to a unique cluster. We consider these two together as one. So 5 external links and this one internal link. So we maximize the internal link, but we also actually increase the external link. So this is not probably a good idea. We go another iteration. We take a different path here. Now we say, okay, what happens if we absorb, pink absorbs orange? So these two, which are closer. This time we count again. There's one external link, and then two here, and these two together, because they are both going to green, 3 external links, and there's one internal link. This is better than going to purple here, because we maximize the internal links before. At the same time, we also minimize the external links. And this process is clear the time. Yeah, this process is repeated until we get we consume. We try all possible ways, and then we just do always maximization of the internal and minimization of external. And then in this manner, in the end, what we get is simply, at the end, you get only 2 nodes, and then we just put them together. So this is kind of hierarchical clustering, what we've seen before. Yeah. And this is one of the most used clustering algorithms in the single cell data analysis, and this in general yields better cluster than the other algorithms. Now, after you know what to do, how to generate your clusters, now let's look at how to find the cell differentiation pathways. There are 2 ways that you can imagine this or view this. One is lineage tree construction, the other one is self-fit inference. The lineage tree construction is about how you can actually generate these mature cell types from this oligodendrocytes. So oligodendrocytes from the oligodendrocyte progenitors, and how can this can be generated from the clear progenitors, and how can this be from the neuronal stem cells. So we want to find out this path here. So this path from how this we can go from here to here. And for this, what we do is to construct a minimal spanning tree starting from here to here. Yeah, that's what we do for the lineage deconstruction. And the other one is self-fit inference. So the cells, for example, here, the neural stem cells can actually choose either this path or this path, one of these, right? They can generate common progenitors or clear progenitors or even neuronal progenitors. So there are three different paths that a neural stem cell can choose from. Yeah, and the decision that cells takes based on its gene expression, maybe we look at the gene expression, how it decides on it, but the decision that it takes to generate one of these mature cell types is commonly called as cell fate. So cell decides to generate a neural progenitors than a clear progenitor. So that's the fate of the cell to generate a neural progenitor. So that's why it's called cell fate inference. In this, that's a common terminology that is used in the single cell. And in this, for this cell fate inference, we start from the stem cells and find all possible routes to mature cell types. So this one, this one, and this one. And then we order the cells, we do a pseudo time ordering of the cells along these trajectories. A trajectory, and then we do ordering, and then based on this ordering, and then we try to evaluate, okay, if there is certain genes that are actually constantly over or under expressed or upregulated or downregulated along this path. So example, maybe you see a gene A, which is always downregulated this path, and then which is constantly, there is another gene B that is upregulated here, and then there's another B gene C, which is downregulated. So if you see a common expression pattern that actually defines this particular trajectory compared to the others. So this is what we are inferring. And there are methods to do this. Yeah, let's first look at how to do this, how to generate the lineage tree. So we are looking at the this part here. So lineage tree construction, we have to look at the global relationships among the cells. So because we want to see how these cells are connected to one another. So we want to find out these parts here, right? And to find this out, we create a minimal spanning tree of clusters. So we don't have these parts, we just have the clusters now. And then start with the clusters, a minimal spanning tree of the clusters. And this minimal spanning tree should reflect the cell proliferation, how these cells generate one type to other. And this a good minimal spanning tree is an indicator of how well the previous analysis, such as quality control and the normalization and the clustering was done. So let's look at how a minimal spanning tree is constructed. So we start with this clustering, then we take the most extreme cluster. This is this blue one here. So we start with the most extreme cluster, and then we connect this most extreme cluster. We group rest of the graph. So we have a connected graph here. We group rest of the graph to the rest of the graph, and then say it as U1, and then our new extreme cluster as E1. And then now we take another extreme point, extreme cluster. In this case, this is, let's say, hello one here. And then we also separate it from the remaining graph, which we put it here. And now for E1, or poses a question that, okay, whether am I close to U2 or am I close to E2? Yeah. If it is close to E2, if the outcome is it is close to E2, we connect E1 and E2 like this, and then we don't have this connection anymore. If it is close to E1, we connect E1 to E2, and then we don't have this connection anymore. So there are two possible outcomes here. And E2 also poses the same question again. And then if it is already connected to E1, and if it poses the question, okay, and do I have a strong relationship to U2, then you connect it. Otherwise, E1 is connected. The E2 is connected to E1, E1 is connected to E2. So we do this. This is already connected here. We connect this here. So this is already connected because this step is already connected. And then we repeat this process again and again until we end up with only one node, one extreme node in this graph, and then we finally have such a minimal spanning tree which preserves the strongest relationships among the clusters, which we can use to infer the lineage of the clusters, among the cell types, the clusters. The next step, what we are doing, looking at, is this one, this fed inference, the self-fed inference. The self-fed inference, we have to find a zero-time ordering of the cells along a given developmental trajectory. Yeah, so we have this, and then we have to find out. We have to find a zero-time ordering of all the cells given this trajectory. And now, one thing that we have to recall that I mentioned previously, that the cells, the stem cells contain higher entropy, that means higher dispersion compared to the mature cell types, because these stem cells tend to give rise to other cell types. So there is no clear expression patterns compared to the mature cell types. This is the assumption here. And yeah, and now what we do here is we start from these types, stem cells, and then going to the less and less clusters, less and less dispersion, and then we do a ring of the cells. This we define anchor cells for each cluster, and along the cells, the other cells are all the cells are placed along this trajectory based on these anchor cells. So we'll see here an example of how we do this zero-time ordering. So we first need a two-dimensional clustering of the cells. So here we have cells 0, 1, 2, 3, and 4. Then based on this clustering and the connections, we create a degree matrix and an adjacency matrix. The degree matrix is calculated as how many other cells or how many other nodes this node is connected to. In this case, 0 is connected to 3 nodes. 1 is connected to 2 nodes. So these connections here, 1, 2, 3 for 0, 1, 2 for 1, 3, 1 and 2 for 3, and so on. So it has only a diagonal matrix with how many connections per each. And there's adjacency matrix. Adjacency matrix, which is generated from the number of adjacent connected nodes to each node. For example, node 0, no, not number of connections, but whether it is connected or not. So if there is a connection between 0 and 1, we put a 1 here. If there is a connection between 0 and 2, we put a 1 here. So there's no connection between 0 and 3, and that's why there is a 0 here, and so on. So the degree matrix, number of connections, adjacency matrix, 1, if the connection exists, 0 otherwise. That's all. It's pretty straightforward so far. And now we create a so-called transition matrix, because we are now going to create a Markov chain. You first create a transition matrix, and then create a Markov chain out of it, and then we do compute the probabilities of this. So we first would start with this step where we create a transition matrix. So the transition matrix, for the transition matrix, we take the inverse of this degree matrix and then multiply with the adjacency matrix here. So this is a simple matrix multiplication, and then what you end up with is this matrix here, 1/6 times this matrix. Then we use this matrix, this transition matrix, to label your transitions in this graph that we are provided with. And this time we don't have a similar symmetric matrix anymore, because when you do a matrix multiplication here, what you get is an unsymmetric matrix. So transition from 0 to 1 is not anymore equal to 1 to 0. For example, 0 to 1, for example, transition from 0 to 1 is 1/3, compared to 1 to 0 is 1/2. So 1 to 0 is here, 0 to 1 is here. So they have different transition probabilities. Let's say we are now creating a Markov chain. We start with time point 0 with the starting probabilities at t0. Let's say we want to visit 0 here, and we initialize with 1 and then remaining with zeros, and we iterate with this condition. So in each iteration, we take, so to compute pt plus 1 for the time point 1, we take the probabilities from here multiplied by this transition matrix. So we do a simple matrix multiplication for each step. So p0, so p1 is p0 times t, p2 is p1 times t, and so on, until at the end it converges where pn times t is pn. So there's no more changes. Let's look at an example quickly, how it is done. So you have this transition matrix, you initialize the p0 with 1 0 0 0 0, and then compute. You do a matrix multiplication, this times this here, and then you get this. Then for p2, you multiply this again with the t, you get this, and so on, so forth, until you converge. Yeah. So after 10,000 iterations, what we see here is, let's say if it is converged already, you see here the probabilities of 0 and 2 are much higher. and these indicates that 0 and 2 are the more connected than the other nodes in the graph. So these are the most, that means these are the most important cells, these anchor cells. Now we start from these cells to order the other ones. So we start from these two and then try to traverse to other cells. And if we get the next cell, which can be traverse be faster, is the next one adjacent to this cell. So let's take an example of 0, because this is one of the cells with highest probability here. So we start with 0. Now we compute so-called commute time. Yeah, the commute time is calculated by the routes where you can reach from one node to other node. So we are here connecting, trying to commute from 0 to 4 first. So we compute the commute time. We take different routes. So 0 to 4 can be directly done. So here the commute time is 1 hops, so 1 step. Can be also 1, 2, 0 to 1, 1 to 2, 2 to 3, 3 to 4. That's 4 steps. It can be also 0 to 1, 1 to 2, 2 to 0 again, 0 to 4. That's 4 steps, and so on. And this, yeah, can be also a longer, many number of steps. Go here, go back again, go forward again, then go down. So yeah, this is just to show an example how it is done, but in the original algorithms that are implemented, yeah, they are always constrained that you should not visit the node that you already visited, or you should not visit the node that you're starting with, and so on. So this is just to show an example how it is done. Yeah, you have how many steps between 0 and 4, all these steps, and then you compute commute time based on average steps. So sum them up, divided by number of paths that you have, and this is what 3.75 is the commute time. And then we compute the commute time for the remaining cells and then order them by this cell. So this is how we order them, all the cells by computing the Markov chains and then compute the commute time. So now you know how to derive the lineage of clusters, how to order the cells along a development trajectory, and now we have to find out what drives this branching points. So branching points are something that is here. Yeah. So a neural stem cell can differentiate into clear progenitors. And at this point here, so a clear progenitus can take this path here, or it can take this path here. One of them, right? So it can go either this way or this way, and then yeah, you have so you, depending on the path that chooses, you have different types of mature cell types. So either astrocytes, or in the end also there can be also again oligodendrocytes if this path is chosen. And now we have to find out what drives this branching point. So what actually is happening here? For this, as I said in the beginning, we have to find the genes. So we have to really look at the expression levels. So what is actually important for taking this decision instead of this decision, right? So to investigate such branching points, we look at a so-called MA plot here. So we have two groups of clusters, and now we look at, okay, an expression, mean expression level on x-axis here. Probably you've seen this for the bulk RNA sequencing, because if you use DE-seq for differential expression, that also produces a similar MA plot. So here you have log2 mean expression. This can be a geometric mean, and here you have log2 fold changes. The fold changes depend how much change, how much the relative, how much is the relative change in expression levels of a gene between two conditions, between this group 1 and group 2. All the red ones here are the upregulated genes at a certain pulse discovery rate, and all the all the blue ones here are the downregulated line genes. So that's why you see log2 fold change, minus here, plus here. Now, then we investigate, okay, what happens, what is happening in the whole transcriptome, or whole, what are the changes in the gene expressions when we transition from group 1 to group 2, 1 cluster. So maybe there is some gene which is very important. Let's say this RRM2, which has to be expressed to take the decision that glia progenitors to produce astrocytes instead of dendrocytes. So these kind of inference we can make out of such MA plot, and then we see find out which genes are actually are important for this branching of the cells. Yeah, so if you want to learn more about the single cell RNA-seq data analysis, you can look at our Galaxy training material. So if you follow this link here, you can find out in the transcriptomics topic. There are lots of tutorials, hands-on tutorials that you can analyze data on Galaxy with some real example data sets. So you can also check out these materials if you want. Yeah. So let's summarize what we have seen so far in this lecture. So we've seen that a single tissue can have heterogeneous cell populations, right? Tissue can produce this, they constitute these purple cells here, or green cells here, or yellow cells here, which have unique expression profiles. And we use these unique expression profiles to find out the most differentially expressed genes, or most highly variable genes among these cells, and then we use this to cluster these cells based on these expression profiles. And we've seen also the cluster is a continuous process, so you sometimes see these hard clusterings where we see well-defined cell types, or it can be also soft clustering where we see the transition of these cells from one stem cells or to really mature, less mature cell types to mature cell types. And from the clustering, we also looked at how we can achieve, how we can build lineage trees to find out which cells are proliferated to which cell types and how the cell differentiation works. We also looked at the cell-fed inference, and also by the branch point analysis, how there are specific some subset of genes that actually decides, or that actually responsible to decide the fate of the gene, the fate of the cell, to generate one cell type than the other one. So that's all for today. Thank you for your attention.